We are going to start with section 4.4 on the nucleus and ribosomes. We are on slide 34, and you can see a lot of terminology on this slide. You can see a lot of words in red, so make sure that you know the definition of all of the terms in red on this page, and I'll go through them with you now. The nucleus of a eukaryotic cell is the command center or control center of the cell. It contains the DNA. It contains the chromosomes, which contain the genes of the organism. So the nucleus is separated from the cytoplasm of the cell by the nuclear envelope, which is also like the plasma membrane. It is or like the phospholipid bilayer of the plasma membrane. It has a double layer. It has a double layer of membrane, which is the same component as the plasma membrane. So double layer of that with pores, so openings, so that pieces of nucleic acids can leave the nucleus and enter the cytoplasm in order to synthesize proteins. So the nucleus is surrounded by the nuclear envelope, and the nuclear envelope contains nuclear pores. Inside the nucleus, when the cell is at rest, when it is not actively dividing, the DNA is in a form that we call chromatin. The semi-fluid gel material inside the nucleus is called the nucleoplasm. And then the chromatin is composed of DNA plus proteins that kind of hold the DNA together. When the cell is dividing, the chromatin condenses into chromosomes. In humans, there are 46 of those chromosomes. They're rod-shaped structures, and each chromosome will contain a certain number of genes that determine our traits, that code for our traits. So another structure inside the nucleus is called the nucleolus. And the nucleolus under the microscope appears even darker than the nucleus. The nucleolus is responsible for synthesizing ribosomes, and then ribosomes synthesize proteins. So we're looking at an image right now of the nucleus of the cell. You can see the nuclear envelope. You can see the pores, the nuclear pores, blown up in a larger detail. Inside the nucleus, you can see the strands of chromatin and the nucleolus. The job of the nucleolus is to produce ribosomes, and then ribosomes produce proteins. Ribosomes are the site of protein synthesis in the cell. Ribosomes may be free. They may just be floating around in the cytoplasm. They also may be attached to the rough ER, the rough endoplasmic reticulum. The way that this all relates and ties in to the central dogma of molecular biology is that DNA, which is found in the chromatin and chromosomes inside the nucleus, DNA contains the code that is transcribed into a structure called messenger RNA. Messenger RNA only contains the code for one particular protein. And then that messenger RNA leaves the nucleus, enters the cytoplasm, and attaches to a ribosome, and it is translated into the amino acids that produce a particular protein. So the central dogma says DNA codes for messenger RNA codes for a specific protein. So really, when we say that DNA codes for our traits, DNA really codes for specific proteins. And the proteins that compose one person are are different from the specific proteins that compose another person, that make up another person. So now in section 4.5, we learn about the role of a cell as a factory. Every cell is like a little factory. It produces some type of product, but usually more than one product. And the component of the cell that produces products like proteins and lipids is called the endomembrane system. And it consists of the nuclear envelope. So you have the outside of the nucleus called the nuclear envelope, which is attached to the endoplasmic reticulum. And then the endoplasmic reticulum is divided into rough and smooth ER. And that both of those interact with the Golgi apparatus, or Golgi. The Golgi packages the products of the ER into these structures called vesicles. And the vesicles transport the products to their destination, whether that be inside or outside the cell. So first, the endoplasmic reticulum. In general, we call the endoplasmic reticulum a system of membrane channels and saccules. So they are channels in which product is made and modified, and that product can be protein. If it's a protein, it's going to be produced in the rough ER, because the rough ER contains ribosomes attached to it. So the rough ER produces proteins and modifies those proteins. Pieces of the rough ER pinch off and form transport vesicles. Those vesicles can move to the Golgi, where the protein is packaged and then sent to its destination in more transport vesicles. The smooth ER does not have ribosomes, and it synthesizes lipids. For example, in the testes of a male, testosterone is produced by smooth endoplasmic reticulum, because testosterone is a steroid, which is a lipid. So the smooth ER, there's lots of smooth ER in the testes in order to produce lots of testosterone. The liver has abundant smooth ER for detoxifying drugs. We already went over that example. So if you look at this image on slide 40, we're starting here with the nuclear envelope, and attached to the nuclear envelope is the rough ER. You can see the ribosomes, the little yellow ribosomes attached. And then continuous with the rough ER, but further away from the nucleus, is the smooth ER. So our proteins are made here in the rough, and then our lipids are made here in the smooth. And the products of the ER are pinched off into vesicles, and those vesicles transport those products to the Golgi. The Golgi actually resembles a stack of hollowed pancakes or pita bread. The Golgi receives vesicles from the ER, modifies the products, whether they are proteins or lipids. The Golgi will modify them and prepare them for being shipped. So they package proteins and lipids into more vesicles that leave the Golgi, and then either those products are transported out of the cell by exocytosis, or those products are transported to some location within the cell. Lysosomes are actually a special type of transport vesicle. They are vesicles that have formed by pinching off from the ends of the Golgi apparatus, and they contain hydrolytic digestive enzymes. They are highly acidic, and they will digest large molecules and break those molecules down into smaller molecules. The lysosomes function in digestion. They function in recycling. In white blood cells, lysosomes engulf pathogens like bacteria. and break them down and destroy them. So a summary of the endomembrane system: proteins are produced in the rough ER, and lipids are produced in the smooth ER. And these proteins and lipids are carried in vesicles to the Golgi. The Golgi modifies and packages these protein and lipid products into new vesicles that transport those products to various destinations within the cell and outside the cell. Some cells will secrete products, and that process is called exocytosis. And this is an image of, and another place that you can learn about the endomembrane system is in your Chapter 4 homework that's posted on Connect. You have, there is a video tutorial on the endomembrane system, and it's really good. So, so here's what we have. We have a vesicle that is forming around this little orange molecule, this orange substance that's coming into the cell. Part of the plasma membrane is pinching off around that orange substance and forming a vesicle. And then we have, in our rough ER, we have some enzymes that have been produced. The enzymes are proteins, and they are being transported by a vesicle to the Golgi. And the Golgi is going to take some of those products and secrete them outside the cell, but it's also going to take some of those enzymes. And the end of the Golgi is going to pinch off surrounding those enzymes. And now we have a lysosome. So a lysosome is just simply a vesicle formed from the Golgi that has digestive enzymes inside of it. So what you see happening over here on the left side is the lysosome fusing with our vesicle that brought in this large molecule. It's fusing with this vesicle, and those enzymes are now going to digest that large molecule and break it down. So the endomembrane system has a lot of different functions, but it's the part of the cell that we like to refer to as, or we like to use the analogy that the cell is a little factory, and that's because of the endomembrane system. Now, in section 4.6, make sure that you know about peroxisomes. They're a lot like lysosomes, but they have a few, they're special. Okay, so one is they really only break down lipids. They function in breaking down fatty acids. And the reason that they're called peroxisomes is that they produce a byproduct called hydrogen peroxide. You may have hydrogen peroxide in your medicine cabinet or in your bathroom somewhere, or even some people use it, they have it in their laundry room because hydrogen peroxide is really, really good at getting out blood stains. So, but hydrogen peroxide is toxic to our cells. So when we have peroxisomes breaking down fatty acids, those peroxisomes are producing peroxide as a byproduct. So they have to also contain an enzyme called catalase that will break down the peroxide into water and oxygen so that it won't be toxic to our cells. So this is showing a plant cell peroxisome under the electron microscope. And then we have structures called vacuoles, which are just larger than vesicles and mostly function in storage. Plants have a special vacuole called a central vacuole. And sometimes if it's filled with water and solutes, it will contain up to 90% of the volume of those cells. So vacuoles, or that large central vacuole, functions in storing water, nutrients, pigments like chlorophyll, and waste products. And in plants, when it fills with water, it helps the plant cell develop turgor pressure, which is what our plants look like after we've watered them. They look nice and healthy, and they're standing up straight and tall and rigid. That's turgor pressure.